Genetics and Genomics and Medicine, Chapter 5, Single Gene Disorders, Inheritance Patterns, Phenotype Variability, and Allele Frequencies. Module 1, Terminology, E-Resources, and Pedigrees. In this slide, we'll discuss some basic terminology that are essential to understanding single gene disorders. As we have learned, genes are functional units of DNA that make products needed by cells, either the polypeptide chains of a protein or a functional non-coding RNA. In this chapter, we consider genes that are responsible for single gene disorders, diseases in which the genetic contribution is determined primarily by one gene locus. Although collectively these diseases are individually rare, single gene disorders are important contributors to disease. Knowledge of single gene disorders also provides a framework for understanding the more complex genetic susceptibility to common diseases, which we will cover later in this course. The first important term to decide is the genotype. The genotype is the combination of alleles that a patient possesses. An individual can be homozygous, heterozygous, or hemizygous. Homozygous and heterozygous are terms that apply to the somatic cells of the body. Hemizygous refers to the condition that occurs on our gametes. If both alleles are the same at an individual locus, a person is said to be homozygous at that locus and is referred to as a homozygote. If the alleles are different, even by a single nucleotide, the person is said to be heterozygous at that locus and thus is referred to as a heterozygote. In our germ cells, although we are essentially diploid elsewhere, men have two types of sex chromosomes, X and Y, each of which are very different in structure and gene content. As a result, most DNA sequences on X do not have a direct equivalent or allele on Y, as would be the case for the rest of our chromosomes. Men are therefore hemizygous for such loci because they normally have only one allele for the Y chromosome. Women normally have two alleles at each locus on the X chromosome, so we can apply the terms homozygous to the female gametes, but we are in a hemizygous state on male gametes. Considering the phenotype or the observed characteristics that correspond to a given trait, we must remember that genetic variation is not the only determinant of the phenotype. Environmental factors can make a contribution too, as well as epigenetic effects, which, unlike genetic mechanisms, are independent of the base sequence of DNA and stochastic factors. And we discussed epigenetic effects earlier in this course. And finally, there is the phenotype. It's very important to become comfortable with this term. The phenotype is the observable manifestation of the genotype. Primarily, the phenotype is due to the sequence of a particular allele, but as we said earlier, environmental factors and epigenetic factors can also affect the given phenotype. So if we're talking about a disease scenario, for example, the phenotype would be the clinical symptoms of the patient that is harboring a particular genetic variation that gives rise to the disease state. Single gene associations. In humans, any genetic characteristic is likely to depend on the expression of a large number of genes and environmental factors. For some, however, a particular genotype at a single locus is the primary determinant, being both necessary and sufficient for the characteristic to be expressed under normal circumstances. Such characteristics are often said to be Mendelian, or those loci are said to be inherited in a Mendelian fashion. That implies that a chromosomal locus is involved. A more accurate terminology is monogenic, which takes into account both chromosomal and mitochondrial loci. When a human monogenic disorder or trait is determined by a nuclear gene, the disorder is said to be dominant if it is manifested in the heterozygote, who carries both a normal or wild-type allele and a mutant allele. Alternatively, one can be recessive if this is not the case. So if an individual needs to be homozygous at that particular allele in order to display the trait, then that inheritance is said to be recessive. Sometimes two different phenotypes that result from mutations at a single gene locus can simultaneously be displayed by the heterozygote and are said to be codominant. For example, if we consider the AB blood group. The AB blood group is the result of codominant expression of the A and B blood group phenotypes that are determined by different alleles at the ABO blood group locus. Electronic databases are available on single gene disorders. Gene Reviews provides excellent summaries for many of the more common single gene disorders that are accessible through the widely used PubMed system for electronic searching of biomedical research literature. The online Mendelian Inheritance in Man, OMIM database is highly comprehensive and can be looked at through using the PubMed system as well. Pedigree symbols. The extent to which a human disorder has a genetic basis can often be established by taking a family history. A pedigree is a graphical representation of a family tree that uses standard symbols, shown in this slide, and is used by geneticists to determine inheritance patterns. Generations are often labeled with Roman numerals that increase from top to bottom. Individuals within each generation are given Arabic numerals that increase from left to right. An extended family covering many generations may be described as a kindred. A family member through whom the family is first ascertained is known as the proband and may be marked with an arrow. The term sib is used to indicate a brother or sister, hence the word sibling. And a series of brothers and sisters is known as a sibship. According to the number of steps in the pedigree that links two family members, there may be classifications of relatives of the first degree, which would include the parent, child, and sibs. Second degree relatives, which include grandparent and grandchild, uncle, aunt, nephew, niece, and then half-sibs. Third degree relatives include the first cousins, and so on. Couples who have one or more recent ancestors in common or are related to one another are said to be consanguous. So sometimes you'll see in the literature the term consanguous marriage, and that would indicate that, for example, cousins are marrying one another. So we can see here from this slide, male is represented by a square, female is represented by a circle, open circle. A diamond refers to sex unstated. When the symbol is unfilled, it means that the individual is unaffected with the genetic condition or phenotype. When it is filled in, and in this particular slide they're showing blue, that color does not have the significance. It's filled in that has the significance. That means they're affected. An individual that has a dot in their symbol indicates that this is an obligate carrier who will not manifest the disease. And then with a line, vertical line, that indicates that the carrier who may go on to manifest the disease, and a line across diagonally means deceased individual. Note how consanguous mating is referred to, mating or marriage, the two that are giving rise to the offspring. Note also how siblings are indicated. So siblings are joined together by a line. Twins have a special denotation, and it demarks whether or not they're identical or paternal twins. And then we have symbols for different types of terminated pregnancies as well.